Welcome to Energy Reactions in the Cell.
Before you begin, a very important warning. Watching this video may cause you to gain some important knowledge which could increase your chances of passing biology at Tulare Western High School. If you do not wish to pass this class, please disregard the information contained in this video or turn off your computer immediately. This video is extremely hazardous to your ignorance. Okay, before we begin, you need to go ahead and review the learning objectives for this particular lesson. Some of this stuff is easy because it's review, but some of this stuff is brand new. You should be able to master all of these things before you proceed to the next lesson. You should be able to, by the end of this video, describe the ATP molecule. You should also be able to explain how energy is stored or released in the ATP and ADP cycle. You should be able to identify 3 compounds associated with electron transfer and show how these compounds could actually carry electrons. You should be able to define heterotrophs and autotrophs. Also, you should be able to define photosynthesis and supply its summary equation. You should be able to explain why chlorophyll is important for photosynthesis and be able to sketch and identify important parts of a chloroplast, including the stroma, grana, and thylakoids. Now, if we're going to have a discussion about energy in the cell, the most important energy molecule in the cell is going to be ATP, or adenosine triphosphate. You can refer to it as ATP, everybody does, but it's important to kind of sort of look at what this thing is made of, because we'll see these basic building blocks again and again and again. The adenosine part refers to a nitrogen compound found in both DNA and RNA called adenine. You don't necessarily have to know the structure of this, but you'll see adenine again as an old friend over and over and over again when we look at nucleic acids. Attached to the adenine is a ribose sugar. It's a 5-carbon sugar, and this 5-carbon sugar is also found in RNA. The other end of the ribose sugar are 3 phosphate molecules, and these are just phosphorus with a bunch of oxygens attached. Now, if we have adenine, ribose, and 1-phosphate, adenosine monophosphate, you essentially have one of the nucleotides of RNA. But in this case, we'll be looking at adenosine diphosphate and adenosine triphosphate with 2 and 3 phosphates respectively. ATP is used as kind of the energy currency of the cell, and we'll see how this happens in just a sec. All right, as it turns out, energy can either be released or stored in the ATP molecule by playing with the bonds that are holding the phosphates together at one end of the molecule. So here's our ATP molecule again, and I'm going to rewrite this to make it a little bit simpler. So we're going to go ahead and take a look at this, and that adenine and the sugar, the ribose sugar, is a compound known as adenosine. So I'll just make a little A here. And then we've got 1, 2, 3 phosphates held together by nice high-energy bonds. And as it turns out, that last bond here can either be broken or formed relatively easily with the right enzymes. So if we want to release energy for cell processes, all we have to do is break this bond, and instead of ATP, adenosine triphosphate, you have adenosine diphosphate, adenosine with 2 phosphates, and of course you've got this little free phosphate over here. Now if we want to go ahead and store energy, all we have to do is go ahead and convert ADP into ATP. In other words, we're going to have to put some energy into forming or reforming that chemical bond here. Thus, if we go from ATP to ADP, energy is going to be released. Note that we're kind of forming a smaller compound, a larger compound here to a smaller compound here. Generally speaking, anytime that happens, you're going to release energy. But the cool thing is that the process is reversible. You can go ahead and take ADP and convert it into ATP, in which case you're going to store energy. Anytime you start out with a smaller compound and end up with a larger compound, chances are you're going to put energy into the system or store it. Now, it's true that anytime the cell wants to use energy, it's going to be using ATP molecules or converting them ATP into ADP. However, sometimes you're just going to want to go ahead and transfer energy from place to place inside the cell. Oftentimes that happens because of the transfer of high energy electrons. And there are 3 important molecules that you're going to need to know. Do not let the names scare you. You're never going to have to go ahead and say nicotinamide adenine dinucleotide or nicotinamide adenine dinucleotide phosphate or flavin adenine dinucleotide, but you should at least recognize the acronyms NADH, NADPH, FADH2. Now what they all have in common, of course, is an H at the end, a hydrogen. So it actually looks like these things are hydrogen carriers, but in reality, a hydrogen carrier is actually going to serve, in this case, as electron carriers. And here's how. NAD, without the hydrogen attached, actually has a positive charge. It exists as a positive ion. Well, normally, if we're looking at hydrogen, hydrogen will ionize, and it also would be positive. So how are we going to get these two together? They're both positive. They're going to want to repel each other. Well, electrons to the rescue, because electrons are negative. So if we add 1 electron to NAD, right, we essentially can go ahead and cause this positive charge to cancel out. That's still not going to be enough to go ahead and attract a positive hydrogen. But all we really got to do is add a second electron. And now, with both of these electrons, this becomes negative, and the hot positive hydrogen is now going to be attracted, and we can have NADH. So even though it looks like it's just carrying a hydrogen, it's actually carrying 2 electrons as well. The same thing happens with nicotinamide adenine dinucleotide phosphate. NADP is a positive 1 charge by itself, so it has to carry 2 electrons to pick up the hydrogen. And FADH, in order to get that second hydrogen, also has to pick up 2 electrons. So in actuality, these compounds are not only hydrogen carriers, but they're electron transfer molecules. And those electrons are going to be really, really important in transferring energy into the cell and around the cell. Are you scared yet? Don't be. It's easier than you think once we see all these things fit together. Plus, hey, here's some quickie review that should be easy. These terms you've had before, and you've seen them a dozen times already, but make sure that they're still a part of your working vocabulary. You should know by now that autotrophs, or producers, can manufacture their own food, and usually they do it through photosynthesis, which we're going to be talking a lot about here. There's another process called chemosynthesis, which we're not going to talk too much about, but yeah, certain bacteria can actually make their own food by just using chemical compounds in the soil. You also know heterotrophs and consumers, right? And they can't photosynthesize, so they have to rely on eating something else to get their energy. And we'll talk about how that energy plays out in a little bit. Likewise, you're probably also familiar with the basics of photosynthesis. That is, that process by which light energy is used to convert carbon dioxide and water into useful food molecules. Down here is a summary equation for it, and you might have already been introduced to this. Light energy, usually in the form of sunlight, is going to be used to go ahead and rearrange the chemical bonds that hold carbon dioxide and water together and build a larger molecule, glucose. And in doing so, we have a little bit of leftover oxygen. Well, we're going to see that all of this is due to energy conversions. And we start out with energy in the form of light and convert it to chemically stored energy in the glucose molecule. Well, there's a lot of other conversions that happen along the way. But before we get into the chemistry behind this, we have to do a quick physics lesson about the nature of light itself. Normally, if we're looking at sunlight, sunlight is just a form of electromagnetic radiation. But it's not quite as simple as just white light. White light is actually a combination of a bunch of different colors, actually different wavelengths of light. And if you combine them all into one, you get white. But you can separate them out, say, in a prism or with a rainbow effect. You guys are all familiar with things like that. But how does light fit into this idea of an electromagnetic wave? Electromagnetic waves or electromagnetic energy comes to us in several different forms based on the amount of energy the electromagnetic wave has and its wavelength. If we have gamma radiation, like maybe released from a nuclear bomb or fusion or fission reactions, they have very, very short wavelengths but very, very high amounts of energy. That's what makes them so dangerous to humans and other life forms. Stretch out the wavelength a little bit, and you still have a very, very high energy wavelength, or excuse me, a high energy form of electromagnetic radiation called X-rays. Give it a little bit less energy, stretch out the wavelength a little bit more, you get in the ultraviolet light, the UV spectrum. There's a very, very narrow band of electromagnetic radiation which we can actually see, we can detect with special sense receptors in the back of our eyeballs, yeah? And this is visible light. The longer wavelengths of light are giving us colors like reds and oranges. Shorter wavelengths of light are giving us things like violets and purples. If we go ahead and continue and have longer and longer wavelengths, we get into the infrared and eventually microwaves and radio waves, but we can't see those. All we can see is the visible light. Well, it turns out that plants make use of capturing certain forms of electromagnetic radiation, and they capture several of the wavelengths of visible light. So we need some special structures for actually capturing light. Special organic compounds that are designed to go ahead and absorb certain wavelengths of light are called pigments. There are several different types of pigments found in nature, but the most important one for photosynthesis is something called chlorophyll. Chlorophyll absorbs pretty much most wavelengths of visible light except green, and that's going to actually provide energy for photosynthesis. Now, you don't necessarily have to look at how chlorophyll actually does this, although it's a good research topic for you. But here's the cool thing. Now, when something appears green, like a leaf, it appears green because it's absorbing all wavelengths of light except green. Green is the one that's reflected out, so green is what you see. Similarly, if you're wearing a blue shirt, guess what? Everything is being absorbed except for the blue wavelengths. And the blue wavelengths are reflected back, so it appears blue. Just kind of a cool thing for, hey, how colors actually work. Now, there's other pigments that are found in plants and leaves. You might find these in lab. Things such as carotenes, phycobilins, and xanthophylls. Think about orange pigments, red pigments, yellow pigments, all those different colors you see in the fall. Right? Now the chlorophyll, at least in plants, is going to be clustered in special organelles called chloroplasts. Chloroplasts are actually green in appearance if we looked at them through a microscope. Here's a view of an electron microscope, and we can see some definite membranes and structures inside. So we're going to take a closer look at these because these are going to become important in tracing what goes on behind the scenes in photosynthesis. So we've got two membranes that are surrounding the chloroplast, but the real cool thing is what goes on inside. We've got several stacks of what look like frisbees stacked on top of each other, or a stack of pancakes. This is called a grana. Each of the little individual discs are called thylakoids, and that's where all the chloroplast is, in the membranes of the thylakoids. We're going to see later that the thylakoids themselves are kind of sort of hollow. In between the grana is not just empty space, it's a gel-like fluid that contains a whole bunch of enzymes that's important for other things, and this is called the stroma. Turns out that different things in photosynthesis happen in different places in the chloroplast. So it's going to be important that you guys be able to list parts of a chloroplast, describe them, and recognize them when you see them again. You also should be able to go ahead and sketch this puppy. Now, you don't have to be an artist to draw a chloroplast. Just give me a little bean-shaped structure and put some discs in there. There's a stack of thylakoids making up a granum, and there's going to be several of these stacks in a given chloroplast. So you don't really have to go ahead and draw this. Just draw something that is recognizable. We can see the stroma. We can see individual thylakoids. If I could write, that's okay, dude. And you can have that stack grana. Now, you're going to be asked to reproduce something like that in exams and quizzes or whatever. But we're not just doing this for the sake of watching you draw. Like I say, different things in photosynthesis happen in different places in the chloroplast, so it's going to be important to go ahead and put a label. Now, you didn't honestly think that we were going to let you get away with not writing about this stuff. So here's a practice quiz for you. It's not really a quiz, but you are going to be graded on it. You do have to turn it in. So respond to each of these. If you have to go back, look the answers up, either in your notes or by viewing the video again, that's fine. But if you're able to do this just off the top of your head, then you're probably prepared for the quiz. If you can't do it off the top of your head, you need to study more. Anyway, best of luck. See you next time.